Okay, so continuing on, we now have tertiary structure. So we talked about primary, which was the actual sequence of those amino acids, secondary, which are these fancy kind of local interactions between amino acids within a peptide chain or a protein chain, forming things like uh, our spirals, which are alpha helices, our beta pleated sheets, or our um, the braid like one. I can't remember the name of it now. Triple helix. There we go. So continuing on from that, these triple helices, these beta pleated sheets, they're not the entire protein. These proteins can be, I mean, they could be 500 amino acids long. Like you're not going to have an entire thing just being one particular type of secondary structure. So tertiary structure is showing the overall structure of your protein. It's stabilized by interactions between attractions and repulsions between R groups of those amino acids. So things that like water, things that are charged, things, we'll go through all of the different ones here. But actually, I'll go ahead and show you this picture. So this is like a cartoon representation of a full protein. So you'll see we start at one end. I don't know which end's which in this particular case, but we start at one end and we see a beta pleated sheet. And then we see just a random chain making some weird shape. And then we see another beta pleated sheet, and then we see another just kind of random chain. And then we see our alpha helices, or alpha helix. So the tertiary structure is how all of this fits together. Erase my highlighting. There we go. So it's overall structure, and it depends on the stabilized by the attractions and repulsions between R groups of amino acids. So there's 5 different particular ones we will talk about. First one is called hydrophobic interactions. So we've talked about hydrophobic things before. These are hydrophobic is things where they like to run away from water. Phobic, remember, is scared of. So they're saying it's scared of water through when you're saying hydrophobic. So non-polar amino acids are going to have this hydrophobic effect or hydrophobic interactions. So the hydrophobic amino acids are going to hide from water. So they're going to be found on the interior of proteins. They're going to try and get as far away from the water as possible. So they're going to go next to another protein, hide away, or next to another part of the protein chain and try and get away from the water that is in the solution around it. Okay, then you also have the opposite, hydrophilic interactions. So philic meaning lover, so lover of water interactions. These are going to be with the polar amino acids. So this is why we have in our chart the separation between the nonpolar and the polar is for these tertiary structures. So these polar interactions, or I'm sorry, hydrophilic interactions between polar amino acids, hydrogen bonding with water, and they tend to be at the surface. So it's the exact opposite. They like being at the surface because that's where all the water is. Next, we have salt bridges. So these are when you have charged amino acids. So maybe you have an ammonium at the end of your amino acid on that R group, and you have a carboxylate. Those like to be near each other because they have, one has positive, one has a negative charge. They balance each other out. So they like to form salt bridges. So these are ionic interactions between basic and acidic side chains. Okay, so the first two are more hydrogen bonding. C, salt bridges, is an ionic effect. D, again, is hydrogen bonding, so just hydrogen bonds themselves. So between hydrogen on a polar R group and oxygen or nitrogen on another amino acid on the backbone, could form hydrogen bonds in interactions to overall make a specific function or specific form. And then the last one we've actually already talked about is disulfide bonds. So these are covalent bonds where you oxidize two thiols in the cysteine residues. So residue, just so you know, is another word for amino acid. So in the cysteine amino acids, they're going to covalently bond to one another. So just as an important little side note, this is the only covalent interaction. So you see the salt bridges was ionic, and then the other three were all hydrogen bonding interactions. So let's practice this real quick. Label the interactions between amino acids, R groups, which help the above protein keep its tertiary structure. So this structure is set. This is how the protein likes to be because of the different interactions. So we're going to try and label all of these interactions. Okay, so this first one here, we have these groups here. So we have, you can see we have our oxygens or alcohols, which are polar groups. They are interacting with the outside of our amino or our protein. So these are going to be hydrophilic. They love to be on the outside. They're polar groups. Oh, I think there's only one L in hydrophilic. Okay, let's go down to the one right below it. So we have this carboxylate with a hydrogen bond to an amine. So specifically this bond here is what we're talking about. So when we have a charged carboxylate and a charged amine, depending on the pH, but two charged amino acids. I'm sorry, this is not the, my bad. This is not the charged amino acids, right? This is just the backbone. So we're showing the amine backbone of the protein. So this is just the backbone. So it's not going to be a salt bridge, my bad. If it were a salt bridge, let's just go ahead and define that real quick. If it were a salt bridge, it would have been an interaction with the actual charge, right? Whereas this is showing the hydrogen bonding to the carbonyl. See the difference? To the carbonyl, not to the oxygen with the negative charge. So the hydrogen with the nitrogen. So this is going to be an H bonding interaction. Okay, so the backbone of our protein chain and a polar side group. In this case, it has just an oxygen or nitrogen that has the ability to be a hydrogen bond acceptor. Okay, so let's go down to this one down here now that I've already messed up. I'm human, guys. But we have this carboxylate. We have that negative charge interacting with the positive charge of our another charged amino acid. These two charged amino acids are going to be forming a salt bridge. Let's go down to the bottom. Okay, so we have two different alcohols. So let's think about what we have here. So it's showing a bond in between the hydrogen and an oxygen, right? So it's again going to be a hydrogen bonding interaction. So we have the hydrogen from one alcohol interacting with the oxygen from another alcohol, forming that H bonding interaction, kind of like we do with water, right? Okay, let's go up to this top one here, top right. Okay, so we have, this is kind of showing these particular amino acids here. So you'll see that we have the aromatic ring with no polar groups on it at all. We have methyl groups. Those kinds of amino acids are our non-polar amino acids. So these are going to be hydrophobic interactions. Also, I just like to point out this, so this is a random coil. Okay, moving on, we have these interactions here where we have our sulfur-sulfur covalent bond in between two cysteine residues. These are our disulfide bonds. And then our last one down here, we just see dotted lines between two beta pleated sheets. These dotted lines represent hydrogen bonds. So the actual sheets themselves, they have their backbones and everything. Those are interacting through hydrogen bond interactions. So that is tertiary structure for you guys.